Hello everybody, you are welcome. In this video we will continue solving the rest of questions in our exam paper 4 variant 42 February March 25 part 2. Here for question number 4, the HFE gene codes for the HFE protein, which has a role in the regulation of iron absorption by the body. Iron is an essential mineral that can be obtained only from the diet. A mutation of the HFE gene known as C282Y causes hereditary hemochromatosis, which is an autosomal recessive disease. Autosomal recessive disease means the alleles carried by autosomes. Recessive disease means this disease occurs when the individual receive two recessive alleles from both parents. Okay, the mutant allele codes for non-function protein, functionless protein. People who are homozygous for the mutant allele, small homozygous for the mutant allele, produce no functioning HFE protein, and this results in an excess of iron being absorbed by the body. This for homozygous. The accumulation build-up of iron absorption in body organs over many years can cause organ damage. People that are heterozygous, capital small, only one allele, one trait carried by the individual in heterozygous individual for the HFE gene do not have hereditary hemochromatosis. Okay? Do not have symptoms of the disease. Just carry one mutant allele. They do absorb more iron from their diet than people who do not have the mutation. Okay? More iron than people who do not have the mutation. Healthy one. But this does not usually have any health effect. Okay? Letter A. Construct a genetic diagram of monohybrid cross to show how two parents who do not have hereditary hemochromatosis can produce a child with the disease. How come? As the parents do not have this disease and their child will have the disease, so both parents must be heterozygous. Both of them are heterozygous. Use the following symbols: H capital for normal HFE allele, H small for mutant HFE allele. Okay, now parents are heterozygous. I mean H capital H small versus H capital H small. We can construct Punnett square like this one. For male gametes, the male can produce two types of sperms. One type contain H capital, other type contain H small allele. For female gametes, the ova of the wife or the mother produce two types of gametes, H capital, H small. 2 times 2 equal 4. So the offspring contain 4 individuals. 2 times 2 equal 4. H capital with H capital, this individual no disease. H capital H small, no disease. H capital H small, no disease. How many with no disease? 3. While the last one in the corner here, H small has hemochromatosis disease. Why? Because he or she receives what? 2 copies of mutant alleles, H small. Okay. Some scientists believe that C282Y mutation may have first occurred in Ireland. Scientists sequenced DNA obtained from 2 human fossil skeletons in Ireland. One of the fossils was 5,200 years ago, and the other one was 4,000 years ago. The scientists concluded that the human living 4,000 years ago did have the C282Y mutation, while the oldest one, the human living 5,200 years ago, did not have this mutation. Explain how analysis of the result of this two DNA sequencing studies could have been carried out to allow the scientists to make these conclusions. Of sure, they depend mainly on stored data in database, okay, as bioinformatics, and compare both the normal gene and also the mutant one. When they compare the DNA sequencing from both two human fossil skeletons with the database stored, they find this mutation and find their conclusion. They can say their conclusion. Look please. Firstly, analysis of fossil DNA. Okay, after analysis and comparing this results with the database in computer system, okay, they find mutation in recent human fossil skeleton and did not find this mutation in the oldest fossil. Okay, by comparing this database sequencing with the database stored in computer system or software programs. Look please, they use knowledge of HFE gene and C282Y mutation sequence. Both are known, both are stored inside database programs. Here, use of database bioinformatics to store the base sequences. Also identify mutation in fossil DNA, identify in which letter, how many letters are different, what are the differences between normal allele and mutant allele, okay, and so on. Also compare fossil sequence with normal one, with normal and with 282Y mutation allele. Okay, now let's define what is meant by bioinformatics. Bioinformatics is a field of study, branch of biology, field of study that combines biology and to facilitate the analysis of large and complex sets of biological data such as genomes, DNA sequences, gene sequences, protein amino acid sequences in different proteins, huge number of proteins, huge number of enzymes. We can store this data, huge data, in database. So bioinformatics field of study combines biology and computer science. Okay. Letter C. At about the same time as the C282Y allele, mutant allele, okay, is thought to have first occurred in Ireland. The lifestyle of people in Europe began to change from hunter-gatherer to farmers. Hunter-gatherer ate mainly meat with some wild plant food. Their diets had high quantities of iron. Early farmers ate mainly plants with some meat. Their diets had lower quantities of iron, and these quantities were often inadequate, not enough, insufficient. Figure 4.1 is a map of Europe showing the percentage of people in different countries who now have one mutant allele of the HFE gene. Figure 4.1 shows the C282Y allele, mutant one, does not occur only in Ireland and is now present throughout Europe. The C282Y allele has been maintained in European populations, even though it is a cause of hereditary hemochromatosis. The question now suggests how the C282Y allele of HFE has maintained in European populations. How maintained in European populations? Firstly, look at the map now. Here's the map. Where is Ireland? This part. Here other European countries. Okay. What about the figure and the percentage occurrence of this mutation in Ireland? 10.1. Okay, near Ireland 8.2, some countries with 8.2, 7.5, and so on. As we go far, move far, okay, to the right or to the north or to the south, the figures decreases gradually. This means what? This means the hypothesis or conclusion of scientists that Ireland is the base or the first place where this mutation takes place is correct. It's correct. It's okay. Now look how this mutant allele has been maintained in European populations. How? Firstly, low iron diet change from hunter-gatherer to farmers. Hunter-gatherers means mainly depend on meat with rich or high quantities of iron. When these citizens shift to become farmers, to eat plant, okay, and little meat, so iron intake become low. Low iron diet acted as selection pressure, okay. Now people with mutant absorb more iron. Just mutant. People with mutant, heterozygous mutant, okay, with mutant allele absorb more iron than healthy one, than normal one, than H capital. Okay. Now advantage. What's the advantage? Advantage for this heterozygous individual with mutant allele, they survive more. They have more survival rate, more reproduction, and they pass their alleles to offspring, of sure. Increase in frequency of mutant allele in population, okay, due to this advantageous selection pressure of low iron intake. Now natural selection occurred. The nature choose the best individuals that cope with the new conditions of low iron intake. Now heterozygous advantage, more iron intake but no disease. Heterozygous individual have no symptoms of hemochromatosis, okay, no disease, no symptoms of disease, but more iron intake, okay. Now mutant allele is neutral in modern diets or offers other protections, okay. Letter I suggests explanation for the differences in the percentage of people in different European countries who have one mutant allele of the HFE gene as shown in figure. Explanation for the differences. Look at this map again, magnified map. Differences in percentage across Europe. Highest in Ireland, okay, the score 10.1 percentage in Ireland. Higher nearby due to migration, 8.1 in adjacent country. Here migration influenced by barriers, borders between countries. Type of diet may differ between regions. Maybe some countries differ on neighboring one by their diets, for example, or food style. What about different treatment access for hemochromatosis? Some countries may present some treatment for hemochromatosis, so the percentage may differ from country to another. It's okay. Now for D, biological databases contain DNA sequences or sequence data from a large number of different people. Table 4.1 shows 3 of these databases, A, B, and C, and the percentage of people in each database who have one mutant allele of the HFE gene. The percentage of people here 2.6, 9.1, 6.3 according to different database A, B, and C. respectively, suggests one reason for the differences between the three databases shown in figure 4.1. Offshore, this due to what? Due to differences in sample size in each database. The information or the data stored in different databases offshore, they are different in size. Also, this sequences for genes of different ethnicity. Ethnicity means races, something like races, different ethnicity, okay, different region of the world. So a percentage of people differ according to sample size, ethnicity of people, region of these countries. Okay, now for letter E. This part is so important. It's about Hardy-Weinberg principle. Letter E, in one population consisting of 2,501 people, there were 9 people who were homozygous recessive for the HFE gene. 9 out of 2,501 are homozygous recessive for the HFE gene. Use equation 1 and equation 2 of Hardy-Weinberg principle to calculate the number of people in the population who are heterozygous for the HFE gene, where first equation P plus Q equal 1, equation 2 P squared plus 2PQ plus Q squared equal 1. Key to the symbols to remind yourself with these letters: P, frequency of dominant allele; Q, frequency of recessive allele; P square, frequency of homozygous dominant genotype, I mean H capital. 2PQ means heterozygous, H capital, H small. Q square means H small, frequency of homozygous recessive genotype in individuals. The question now, he asked about calculate the number of people who are heterozygous for HFE gene, equal what? Firstly, from the data given, 9 people who were homozygous out of 2,500 and 0. This homozygous recessive people means frequency of them equal what? Equal, look please, here, number of homozygous recessive individual equal 9. Frequency of homozygous recessive, I mean Q square, equal 9 over 2,500 and 0 equal 0.0036. What about Q? If we need to obtain Q equal the square root of this figure, then Q equals square root of Q square, I mean Q equal square root of 0.0036, I mean 0.06. Okay, now we obtained Q. What about the first equation of Hardy-Weinberg? P plus Q equal 1. So we can obtain the value of B. How come? Where B plus Q equal 1? So P equal 1 minus 0.06. The result equal 0.94. That's for P. Now P obtained. Okay, we can get B. Okay, we got B and got also Q. What about heterozygous individual? It represented by 2PQ. 2PQ, I mean 2 times H capitals times H small. Here, look, heterozygous for HFE, 2PQ equal 2 times the value of P times the value of Q equal 0.1128. This is the value as frequency. frequency of alleles in gene pool of heterozygous of individual in this population that formed from 2501. What about the real number of individual affected or carrying this genotype H capital H small? How many out of 2501? How? So we have to multiply this frequency times what? 2501 to obtain 282 individuals. They are heterozygous H capital H small. Okay. Question number 5. Figure 5.1 shows photomicrograph of single plant cell in stage of meiosis. Describes the stage of meiosis shown in figure 5.1. Please believe in your eyes. Here two cells. These two cells are joined by, they are plant cells, so they are joined by middle lamella, not complete cell wall. Okay. Here two cells. Each cell consists of two groups of chromosomes, but each group only sister chromatids. They align not at equator, away from equator, further from equator of the cell. So this phase is anaphase 2. Two cells means meiosis 2, starting of meiosis 2. Okay, here two groups of chromatids, two groups of chromosomes in the form of chromatids, okay, separated away from each other. Okay, so this phase is called anaphase 2. Look at this video. Here two cells. Okay, after metaphase, anaphase, where sister chromatids separated by spindle fibers or microtubules towards each pole of the cell. No nuclear envelope surrounding each group now. So it is not telophase 2, it is just anaphase 2. The answer now, here meiosis 2, two cells visible, suggests second division. Okay, stage anaphase 2, the stage, precise stage anaphase 2, where each cell shows two groups of chromosomes moving apart towards the pool of the cell. Here centromeres attached to spindle fibers, microtubules. Okay, spindle fibers contract, shorten to pull chromatids towards the pool. Now chromatids pull to opposite pools. Here centromeres lead with arms trailing, bending or trailing. Okay, this confirm the stage is anaphase 2. No nuclear envelope, confirm that is not telophase 2, it is anaphase 2. It's okay. The video for the last time, step by step. Look, starting from here. Look, after at the end of metaphase 2, centromere split into two centromeres, each centromere for each chromatid, for each sister chromatid. Then the spindle fiber pull or contract to pull the sister chromatids away from each other towards the pools of the cell. Look. 6a, figure 6.1 outlines part of the control mechanism that regulates blood glucose concentration. Meal rich in carbohydrates leads to increase in blood glucose concentration above the set point. Pancreas releases P. P stimulates glucose uptake in fat cells and Q cells. Also P stimulates liver cells to convert glucose to R. So both sides leads to decrease in blood glucose concentration. This figure 6.1. The question now, identify letters B, Q, and R. For B, pancreas secrete P when we have a meal rich in carbohydrates, when we have increase in blood glucose concentration. So B is insulin. B is insulin hormone. What about the left side? B stimulates glucose uptake in fat cells, adipose tissue, and Q cells. Q cells may be muscle or liver. Insulin stimulates the liver and the muscle to store more and more glucose, okay, to glycogen. Store glucose and convert glucose into glycogen. Stimulate both cells to uptake a lot of glucose units. For B here, stimulates liver cells to convert glucose to R. Letter B, okay, stimulate glucose to convert glucose into glycogen, so R glycogen. Now P insulin, Q muscle or liver or both, no problem. For R glycogen, okay. Now letter double I. State the type of homeostatic control mechanism operating in figure 6.1. Here, as blood glucose level increase after having a meal, then decrease after the action of insulin hormone, okay, this and reach and return back to set point. This called negative feedback mechanism. Most of our control system done by hormones in our body done by negative feedback mechanism, okay. For letter B, when the blood glucose concentration decreases below the set point, a hormone is released from the pancreas. Describe how the release of this hormone leads to the blood glucose concentration returning to the set point. How come? Blood glucose concentration decreases when we are fasting, okay, for long period of time. It may reach below than 70 milligram per 100 ml of blood. That's below set point. But our pancreas respond to low blood glucose level by certain cells called alpha cells of pancreas. Okay, sense this lowering blood glucose concentration and start to release what? Release glucagon hormone. This hormone is glycoprotein. It is peptide hormone. It is directly passed in bloodstream like insulin. It circulates in blood, but has no effect on all cells except what? Except liver cells, hepatic cells. Why? Because the cells of liver, cell membrane of liver, hepatic cell membrane contain receptor proteins, specific receptors, complementary fat with this peptide hormone. The peptide hormone is the first messenger, okay, pass from blood capillary to tissue fluid and received by hepatic cell with complementary fat to its surface. This combination, this binding trigger certain protein called inactive G protein, this purple in color, okay, to become active. This G protein, activated G protein, is a mobile enzyme within the membrane. It moves along the membrane and trigger certain enzyme called adenyl cyclase enzyme. This adenyl cyclase enzyme has a role in converting ATP into a compound called cyclic AMP, adenosine monophosphate. This considered second messenger. This molecule is small and soluble, okay, and easily can trigger amplification and response. okay, can make amplification of the signal coming by peptide hormone from outside. The peptide hormone, as we know, it is hydrophilic soluble glycoprotein. It doesn't passes the membrane, but only knock on the door, knock on the receptor protein, trigger the G protein. It doesn't enter the cytoplasm. It is not steroid, it is peptide hormone. So now cyclic AMP act as second messenger and trigger sequence or series of enzymatic reaction called enzyme cascade reactions, like pieces of dominoes. Okay, this reactions leads to activation of certain enzymes. At the end of this series, certain enzyme called the glycogen phosphorylase enzyme is activated now. This enzyme responsible for hydrolysis of glycogen in process called glycogenolysis, or breakdown of glycogen into small molecules of glucose, okay, monosaccharides. This diffuses or passes to outside of the cell in the bloodstream of the liver, okay, through the bloodstream surrounding the liver in hepatic vein. It's okay. Now look at this video. Okay, here's the hormone. It's peptide hormone received by specific receptor. The receptor triggers certain inactive protein into active by using certain kind of energy called GTP. G protein is mobile along the cell membrane of hepatic cell, activate adenyl cyclase to convert ATP into cyclic AMP. This second messenger that creates enzymatic cascade reactions leads to formation of certain essential enzyme called glycogen phosphorylase enzyme that break down glycogen into glucose units. Okay, here glucagon mechanism. Glucagon released by pancreas by alpha cells of Langerhans, binds to receptors on liver cell membrane, okay, then activates G protein. This G protein activates adenyl cyclase enzyme. Adenyl cyclase enzyme converts ATP into cyclic AMP that considered a second messenger responsible for magnification or amplification of the signal. Triggers enzymatic cascade reactions, leads to production of enzymes, phosphorylated enzymes like glycogen phosphorylase enzymes, the last one. Okay, here signal is amplified by the action of cyclic AMP. Glycogen is broken down into glucose unit in process called glycogenolysis. Okay, lysis of glycogen, breakdown of glycogen. This is called glycogenolysis. Now also glucagon. Okay, after glycogenolysis, glucose increase in hepatic cell and release into bloodstream. What about other effect of glucagon? It also act upon adipose tissue or fatty tissue or protein content and stimulate conversion of lipid into glucose and protein into glucose. Lipid and proteins changed or converted into glucose. This process called gluconeogenesis. NEO equal NEW equal new. Gluconeogenesis, formation of glucose from new sources other than carbohydrates. Okay.